come in early and it still doesn't work. Drives me crazy. All right. So no iClicker today. No iClicker today. It's not working. So we will work on that. I don't know what to say about that. It's not even a Monday. All right. The other thing I wanted to tell y'all is that I did update your iClicker grades in Moodle so that you could kind of see how that would affect your grade. Now, it's not one of those things that I can just pull very easily. I have to go in and manually pull them. So I'm not going to do it very often. But you can figure out your grade if you know what your iClicker score is, because there's something in that category now, right? So a 70% is like getting 150 out of 150. Okay? So if you need help calculating, you don't understand whatever it is, let me know. But the calculation is in the syllabus. It tells you how to calculate that 100 and how many points you get out of 150. Okay. Then a couple of people were asking me for more, let's go back one, for a little more detailed, like, can we do this a little more slowly, converting from a straight chain to a ring? So I went ahead and made a tutorial and put it into the announcements, and I put it into the chapter 6 folder. So, you know, in case you want that. But we'll kind of work on it a little bit more today. So we're going to try to finish chapter 6 today. If we don't quite finish chapter 6 today, it's okay, because chapter 7 isn't as long as chapter 6 is. So we'll have time to pick it up. So you can count on our exam date not changing, but we will cover everything. But we're not going to rush. It'll just kind of work out. Okay, so do y'all remember a long time ago, when we were talking about oxidation and reduction? What is oxidation? A loss of electrons. What else? A gain of oxygen or a loss of hydrogen, right? So whenever you get your exam paper, you're going to write that down. And then if you have a reduction, reduction is gain of electrons, loss of oxygen, gain of hydrogen. You need to be able to recite that to yourself and write it down. You have to. So if you have an aldehyde, what's an aldehyde? Remember, an aldehyde is all alone. So it only has one R group. So what else is attached to that carbon now? You have a hydrogen, and then what else? Double bond oxygen. What do we call this carbon in a double bond to the oxygen? What do we call that group? A carbonyl carbon. Okay. So when an aldehyde undergoes oxidation, what's going to happen is that you are actually going to add an oxygen to that. So if you undergo oxidation, what's going to end up happening is that you keep that carbonyl, but you end up adding an additional oxygen where you only had a hydrogen. So where your old hydrogen was, now you have a alcohol, a hydroxyl group, right? So what do we call something that has an alcohol that's attached to a carbonyl carbon? We call this a carboxylic acid. Now, an aldehyde, if you go the other way, if you reduce it, when you reduce something, you're adding hydrogens to it. So when you reduce it, you start out with this carbonyl, you had your R group and you had your hydrogen. Well, you can't just add another hydrogen without changing the bonds, because how many bonds does carbon have? It has 4. So what happens is you add a hydrogen down here, and then that breaks one of the bonds in the double bond. And so you end up with a single bond to the oxygen and a hydrogen. So you do end up also making an alcohol here. So this is a carboxylic acid. This is an alcohol. If you remember what oxidation and reduction are, you don't have to memorize this. You just kind of have to keep track, right? So when an aldehyde undergoes oxidation, so here's our aldehyde undergoing oxidation, you're going to get a carboxylic acid. If the aldehyde undergoes reduction and we go the other way, we're going to make an alcohol, right? Okay, so we had to draw some of those. So let's draw some, right? So the sugar alcohol erythritol is often included in low-calorie sweeteners. It's 70% as sweet as sugar. Erythritol is the reduced form of the aldotetrose erythrose. Oh my gosh, right? But it's a lot of big names, but it's not a big deal. It's really not a big deal. Okay, so what I want to do is I want to look, this is the reduced form. I could write, oh, technology today, you are not my friend. Why is the click dragger working and the pen is not? Oh, the humanity today. Okay, I'm gonna have to restart the computer. When that happens, it's just like the pen doesn't want to communicate, and it means that the computer hasn't been restarted in a while, and it always ends up happening to me. So if you are on Zoom, stay on Zoom. I will be back. Do not leave. All right, I want to do power. We're going to restart. Okay, I'm unmuted. We're going to go back to chapter 6. Note to self, always check to make sure the pen works before you try to start. I don't want to recover lost files. No. Okay, so what are the, how does, wait, I'm like, I'm not prepared to tell this joke. I got to think about it for a second. How did that go? Because if I'm going to tell that, I'm going to give away the punchline if I do it wrong. It's what are the weakest days of the week? That's not right. It's got to be a different word than week. What are the least strong days of the week? The weekdays. I know I really messed that up, but there's another word for week, and I can't think of it right now. It's gonna bother. That is really gonna bother me. Okay. Yeah, the pen works. Okay, let's see. Oh, maybe somebody in the chat's giving me one. No, you did not miss the attendance. The iClicker app is giving me lots of problems. So I'm going to try and uninstall and reinstall and get on the phone with IT this afternoon because it was supposed to be fixed last time, but it's not. So I'll be on with IT. So don't worry about it today. Okay, so we have our sugar alcohol erythritol, right? That's what we want to make. Do we have an alcohol here in this drawing? Well, we have some alcohol groups on there, right? But we don't have erythritol. because erythritol is the reduced form of the aldo tetrose erythrose. And that's what this is. That's what this is. So we need to make an alcohol version of this, which means that we are going to change what's around our carbonyl carbon, right? So how can we do this? What is it to reduce? What are we going to do? We're going to be adding hydrogen, and usually hydrogen exists as a diatomic form, but you don't have to particularly know that to make your products, right? Okay, so everything is going to be the same that doesn't have to do with our carboxylic acid group, with our carbonyl group. Okay, so now here's our carbon. We know we're going to have to keep our carbon. We know we're going to keep our oxygen, and we know we're going to keep our hydrogen. But something else is going to have to change, right? So what happens when we add hydrogen? We're going to break one of these double bonds. So we're going to break, B-R-E-A-K, break one bond. So I'm going to end up with only one bond connecting my carbon and my oxygen. But is carbon happy? Why not? He doesn't have a complete octet. He needs 4 bonds to have a complete octet. So I'm going to have to give it a bond, right? So now we have 4 bonds, 1 down to the carbon below, 1 to the hydrogen, 1 to the oxygen, and now we have an empty one. What is the only atom that we can add? Hydrogen. We can only add hydrogen. Is everybody happy now? Nope. Who's not happy? Somebody's not happy. My oxygen that was in the carbonyl is not happy because it only has 1 bond. What does oxygen want? It wants a complete octet, but does it need 4 bonds? No, it only needs 2 because it has 2 lone pairs. So what am I going to do? I'm going to give it a hydrogen bond, right? Okay, that looks good. Like that is structurally correct. You will not see it like that in your textbook. That's showing all the bonds. And us chemists, you know, we're lazy and we like to show it in as few things as possible, right? So how can I write that stuff in black, but in a more condensed form? So I have my carbon. It has what 2 things attached to it? CH2, right? Then what's attached to that? An OH. And then you can just put in your other groups that I just kind of didn't worry about. So these 2 things are the same thing. Either one of these 2 things that I drew is exactly the same thing. Okay. You good on that? All right. So then I think this one we did. So now we got to move over. Oh, and make sure we're going to do some more in a minute. But if you need more practice doing these, go to that tutorial. It helps you do it because we did some in class last time. I did some on the video, but I made an additional video in case you need some extra practice. So, and we'll do some when we do it today. All right, so we know about simple individual monomers of a sugar. We know how to turn them into a ring. But is that how they normally exist a lot of times inside of us and inside of plants and things like that? No, they usually exist as disaccharides, oligosaccharides, polysaccharides. So how do you go from a monosaccharide to a polysaccharide or any of the other ones? You connect the individual monosaccharides by glycosidic bonds. So it's just a glycosidic bond, it's just a bond between 2 sugars. So when this happens, it fits into a general category of reactions that we've already talked about. So if you have 2 monosaccharides that are reacting together, you are joining 2 things. Do you remember this? Plus ROH, what did we make? you remember this? R, O, R, and what else did we make? Water. What kind of a reaction was that? Careful. What did we make? We made water, so it's condensation. It's that water on the outside of your glass, that condensation, right? So if you are making water, what are you doing? You are joining molecules together, right? That's what happens when you go from 2 monosaccharides to a disaccharide. What happens when you do the reverse? When you go from a disaccharide, what are these R groups in this case now? These are sugars, right? One R is a sugar, one R is a sugar. So what happens when you have a disaccharide and you want to go the reverse? You want to break them up. What else do you break up? You break up the water. And one of them gets an OH and one of them gets an H. So what do we call that kind of a reaction? Hydrolysis. So remember when I said all this stuff keeps coming back and keeps coming back? It keeps coming back and coming back. Okay. So if you are undergoing a condensation reaction, you're making water. If you are breaking water, you are undergoing hydrolysis. So 2 monosaccharides making a disaccharide is condensation. Another way to say 2 monosaccharides reacting to form a disaccharide is the formation of a glycosidic bond. That's condensation. That's the definition, right? Okay, so let's try a few other ones. A disaccharide breaking into 2 monosaccharides. What do we got? Hydrolysis. Okay. A reaction in which 2 molecules combine, forming one, and a molecule of water is produced. Condensation. The breaking of a glycosidic bond. Hydrolysis. Good. Okay. So when we look at a disaccharide, here's an example of a disaccharide, right? How do you know it's a disaccharide? We see our 2 ring structures. And what do we see connecting the 2 ring structures? This is indicative of a sugar glycosidic bond. This oxygen right here, right? So this is telling me that I have a glycosidic bond. Whenever we're looking at disaccharides, a lot of times their chemistry, how they behave, has to do with how those 2 sugar molecules are linked together. So what we try to do is to look at them and then classify them and say, what kind of a linkage is that? It's a glycosidic bond, but what kind of a glycosidic bond? Because it's going to give us tons of information. So you have to be able to look at a sugar molecule and say, what kind of a bond is that? And then it'll tell you all kind of properties about the different sugars, which is what we're going to do in the very next thing. So when you're trying to label what kind of a glycosidic bond, the first thing that you do is to look for the anomeric carbon. What's the anomeric carbon? We talked about it last time. So it's a carbon that's between 2 oxygens. Now, one of those oxygens is in the ring, right? So one of those oxygens is inside the ring. Where's the other oxygen? Down or up, right? It could be there. It could be, whoa, supposed to be erasure. It could be here, right? Depends on if it's cis and it's beta, or if it's trans and it's alpha, right? So in my molecule, what I want to do is I want us to number the carbons, and then we're going to use the numbered things so that we can talk about the different carbons. So in the top ring, what number is this carbon? Carbon 1. Are there times when that carbon will not be carbon 1? Yes. if you have another carbon, CH2OH, something else coming off of it, this would be carbon 1. But we don't have that yet. You will. I keep saying you will, right? So that's carbon 1. Now, in this case, carbon 1 is the anomeric carbon because it's bound to 1, 2 oxygens. Carbon 1 is not always the anomeric carbon. Most of the time it is, but not always. So be careful with that. Okay, then we're going to go around the circle, go away from the oxygen. Remember how when we made those rings, they were particular carbons? So you have to go away from the oxygen. So 3, 4, 5, and then 6 is sticking out of the ring. 6 is always going to be out of the ring, and it's always going to be what? Up. Always up. Okay. So now I found, this is my anomeric carbon. Where's my anomeric carbon on the other ring? An OH. That's my anomeric carbon. So let me number it. 1, 2, 3, 4, 5, 6. Does my anomeric carbon on the second sugar participate in the glycosidic bond? No. Only the anomeric carbon on the first sugar participates in the glycosidic bond. That's going to be important later. Okay. So what I want to do is I want to be able to name this glycosidic bond. So the way that I do that is I tell what carbons are participating in the bond. So list always left to right. So on the left-hand side, which carbon is participating in the glycosidic bond? 1. In the second ring, which carbon is participating in the glycosidic bond? 6. So we call it a 1,6. But that's not quite enough. We also need to talk about the orientation of the glycosidic bond according to the ring. So remember how we said that this guy is up, right? So if the glycosidic bond is up, it's beta. If the glycosidic bond is down, it's alpha. So look at the structure. Is the glycosidic bond up or down? It's down. It's going downward. So is it cis or trans? Trans, and trans in sugars, we say alpha. So this is an alpha 1,6. Now you could just have written in your notebook alpha 1,6, but you have to be able to go through these steps and do it again. Okay? So we're going to practice. We're going to practice. All right. That was the end of that page. Let me go to the next page. All right. So for each of the following disaccharides, name the glycosidic bond and draw the monosaccharide units produced by hydrolysis. Okay, so let's take it one at a time. Let's name it first. What are we going to look for first? My anomeric carbon. So my anomeric carbon is between 2 oxygens. Here's 1 oxygen, here's another oxygen, right? So that's my anomeric carbon on one of them. Where's my other anomeric carbon? In the what? Right here, right? Because I'm bound to 1, 2 oxygens. It's that same oxygen. Okay? All right. So now I'm going to go in and I'm going to start numbering. Let's number the top ring first. The anomeric carbon is carbon number 1. Okay, so go around away from the oxygen. 2, 3, 4, 5, 6. Okay, let's do the same thing below. The anomeric carbon is carbon number 2. This is carbon number 1. Remember how I said it was going to happen? It happened. Okay, now go in the ring away from the oxygen. 3, 4, 5, 6. Now, do we think we can name it? So tell me the numbers first. 1, it's a 1, 2, put it in parentheses, and now we have to put on the outside of it, if is it alpha or is it beta? Okay, it's a good, y'all are really good. So what is this? This is up, right? What is this? Down. So up, down tells me what? Up, down tells me trans, which is alpha. But what's weird about this one? We have another anomeric carbon that's participating in the glycosidic bond. If you only have one anomeric carbon that participates in the glycosidic bond, you only have one of those weird alpha beta symbols. But what do we have? We have two. We have two. So we have to label them both. So now I'm going to look and I'm going to say, here's my up. And what's it doing? It's going up. So this is beta. So it's alpha beta 1, 2. Y'all got this. Y'all got this. I know y'all do. You got it. You got it. Okay. Now we have to hydrolyze this. What are we going to do? Can I erase some of the stuff that's all over it so that it's a little bit easier to see? Y'all okay with that? Okay, I'm going to leave the, I'll leave the number up, the label. Okay. So if we're going to hydrolyze this sugar, what do we have to do? What kind of a reaction? What are we going to add? Hydrolysis. We're going to break up water. So H2O, I could also write as what? This is how it makes more sense to me for these. Instead of writing H2O, to write HOH. Because I know one of the molecules is going to get an OH, and one of the molecules is going to get an H, right? You just got to decide who gets what. So where are we going to break it? Glycosidic bond. So basically, you can draw a cut site either above or below the oxygen, does not matter. So I'm going to draw one right here. This is where I'm going to cut my molecule. And so whichever one got the oxygen, this one will only get a hydrogen. Whichever one did not get the oxygen gets the hydroxide. Yeah? So now we just redraw our rings. What do we got? We got an OH going down. We got a CH2OH. We got an OH going down, right? Okay, so now I need to add what's going to be there. So I picked up an OH from water. That's this one. So now I do the same thing down here, except draw your sugar correctly. Right? This is a CH2OH. This is an O. Move that. Oh, and Jordan, I'm sorry, I'm going to read your note in just a second. I didn't see it. OH, and I need a CH2OH. Okay, now, what do I need to add here? Not just a hydrogen. I need to add the hydrogen, but don't forget this one. You already had an oxygen there. A lot of people just forget that oxygen and then put a hydrogen in and go, I'm done. Yeah, so we're going to try not to make that mistake, right? And put that oxygen. So that's why I draw them in different colors, so that you can see that you kept this oxygen, that's the black oxygen, and you picked up just the red hydrogen. So if you need to like do your homework in colors, that's cool too. How do you know that both are participating? Only one did last time. Okay, so how do you know they're both participating? Well, you have to go in and find your anomeric carbons, right? So when I'm looking for my anomeric carbons, I'm always looking for a 1 carbon that is bound to 1, 2 oxygens. So here, 1, 2 oxygens. So if we go back to our previous one that we did, which was here, right? Here's our 1 anomeric carbon because it's participating in a bond with 1, 2 oxygens. Over here, I'm only participating in 1 oxygen bond. So I don't have to name it because it's not actually participating. Does that make sense, Jordan? Come down. This is where I left off. Yes, good. Awesome. Okay. So let's do the same thing in B. Where is my anomeric carbon in B? First thing I look for is my anomeric carbon. Right here, I'm participating in 1, 2 bonds with oxygen, right? How about in my other carbon? Where's my anomeric carbon? The other side. So 1, 2. Okay, so I found my anomeric carbon, so I'm going to kind of erase this. That way I can see. Okay, now I want to number my carbons. So my anomeric carbon is number what? 1. So then just go away from the oxygen. 2, 3, 4, 5, 6.
So you always, so every time you find an aromatic carbon, you always put the weight knob. Is that like a rule, or do you?
When you're numbering the carbons, you find your anomeric carbon. Your anomeric carbon is typically carbon 1 or carbon 2. And then when you continue numbering, you go away from the oxygen, absolutely, because of how the ring formed, right? Because we had to keep track of that last time, remember? We were keeping track of where our carbons were. And so they have to go in this ring because this is the carbon that's at the bottom of the chain. Remember when we had these and it ended up, so there's my carbonyl carbon, right? And I had a CH2OH. This CH2OH is that CH2OH. So you have to keep the numbering. Yep. Okay. So erase this so we don't get confused. Okay. Now numbering here, my anomeric carbon here is carbon number 1, 2, 3, 4, 5, 6. So there's no other carbon that's coming off of here. If I had a carbon coming off of here, CH2OH, this would be carbon 1 and this would be carbon 2, but I don't. So that anomeric carbon is carbon number 1. Okay. All right. So what are my numbers? What number should I put for the sugar on the left? 1. How about the number for the sugar on the right? Which one is participating in the glycosidic bond? 1 and 4. I intentionally put my cursor over here to see if I could trick y'all. You got to be careful. Be careful. What's that? Well, as long as it works here and not on the exam, we're all good.
So one more time.
Uh-huh.
Why is it, so you have 2 oxygen in there.
These two?
Okay, so at 4 on the second.
Uh-huh.
So why is that not your anomeric?
Okay, so this carbon is bound to 1 oxygen, bound to a carbon, bound to a carbon. And what does that tell you? You must also have a hydrogen. So this carbon is only bound to 1 carbon.
Okay, I was seeing that.
Yep.
OH.
Yeah, the OH here is bound to carbon number 3.
Right.
Yep. So it's all about finding, when you find the one that's bound to 2 oxygens, that was the one that was in the carbonyl bond. And so you have to start your numbering up there.
And so you're gonna have, like, you can have the beta or the alpha if there's 2 carbons on the.
If you have 2, if the 2 red dots are both participating in the glycosidic bond, then you're gonna have alpha alpha, alpha beta, beta alpha. You'll have 2. But see how here we only have 1 dot participating, then we're only gonna have 1 symbol.
And is that always going to present over under for alpha beta?
Not always. Not always. But I think your book is pretty good about that. It's always going to show it to you in 3 dimensions instead of the lazy way that other chemists do it. So I think your book does it always one on top of the other if it's that way. Okay, so are we alpha or are we beta? Because this one is up. And what direction is that glycosidic bond going? Up. So what are we? Cis or beta? But now we got to break it up, right? That was part of the problem. Now we have to break it up. So we're going to add water. But but I'm trying to remember to write it like this. HOH. So where do we want to break it? at the glycosidic bond, so either on the top or the bottom or the left or the right of that oxygen between the two sugar molecules, doesn't matter which one. So I'm going to draw it right there. So this one hydrogen is going to go to the molecule that has the oxygen. This OH is going to go to the molecule. It's going to connect to carbon 4. Okay, so now we just have to redraw them. So oxygen, we got our OH going up. We got a CH2OH. Now carbon 4, right? So this is carbon 1, 2, 3, 4. Carbon 4 was the one that was participating in the glycosidic bond. So carbon 4 needs a downward OH. Is it important that direction? Absolutely. Absolutely, because you did not break the bond. So you need to make sure you didn't break the ring. So you need to make sure it's on the same side. Okay, so that's one of my molecules. So my OH from water was right there. So then I'm going to draw my other molecule. Right? CH2OH, lots of drawing for this class. And I'm not the best artist at all. Okay, so then I have my oxygen, and so my oxygen is just going to pick up a hydrogen. Yes, ma'am. You will not have to draw them, but you will have to select the correctly drawn one. Which is the same thing. I mean, I can't hand grade that many, you know, and get them back to you in decent amount of time. So you will have to pick them out of a set of them. But if you draw it, it'll be quicker if you just draw it on your scratch paper and then compare it to the answer choices. Yeah. Do I need to move up, down? Can y'all see? Got it all?
Yes.
So the direction after we get that glycosidic bond, because it's going up. The first guy, he's going up, so he stays up, and the second guy is
It's going down.
Stays down.
So he stays down. That's exactly
That's
right.
what I was getting
Don't change
at.
him up.
That's what I was getting at.
Don't change them up. You know, you can't invent things, right? You have to take what you have and make it work. All right. All right, so now we can kind of talk about some disaccharides. So identify the disaccharide that fits each of the following descriptions. So ordinary table sugar. If you like to add sugar to your coffee, what kind of sugar are you actually adding? Sucrose. C-R-O-S-E, sucrose. In milk and milk products, lots of people say, oh, I'm lactose intolerant, right? So that's lactose. L-A-C-T-O-S-E. And then the older we get, the more lactose intolerant we get. There are a few people who live in certain parts of, oh goodness, I'm trying to remember the name of the country, I don't know which one it was. But they have been basically living off of cow's milk for thousands of years. And so they don't lose the ability to metabolize lactose like most people do. So as you get older, most people lose that ability. Okay, also called malt sugar, maltose. Okay, now what is the name of the disaccharide that once you hydrolyze it gives you galactose and glucose? Lactose. So lactose is equal to galactose plus glucose. Okay. See, we're doing good. All right, so y'all know the reason why companies use fructose, high fructose corn syrup instead of sucrose? No, but it's on everything, isn't it? Look at the sweetness, the relative sweetness. They've made sucrose basically 100%, right? So if table sugar is sucrose, That's kind of like our standard. And everything is either sweeter than sucrose or not as sweet. If it's sweeter, it has a number greater than 100. If it's less, it has a number less than 100. Okay, so look at fructose. Look at the amount of sweetness fructose has. 50% more to 75% more. That's a lot. So guess what manufacturers can do? They can use less substrate, less product, and make their candy bar taste just as sweet. So it's a cost thing. It's a money thing, like everything else in this world, right? Okay, so if we come down, right? Let's see. Okay, if one sweetener packet of Splenda, sweet and low or equal, has the same sweetness as 2 tablespoons of sugar, according to our table up above, which of the packets contains the smallest amount of sweetener? Now, each of your individual artificial sweeteners are sweeter than normal sugars, right? So we set sucrose to 100%, right? Here's sucrose, 100. These are off the chart, right? Splenda, sweet and low, and stevia, all off the chart. Orders of magnitude sweeter, right? So the question is, if we put enough product into a packet, right? You know, that little pink packet, the little blue packet, not everything in that blue and pink packet is actually artificial sweetener. A lot of it's filler, so that it looks like 2 tablespoons, because that's what we like. We are creatures of habit. If I want 2 tablespoons of sugar in my coffee, I want 2 tablespoons of Splenda. If I decided to do equal tomorrow, I want 2 tablespoons of equal, right? This is how we are. This is humans. So the question is, in which of these packets would be the smallest amount of the artificial sweetener? So which one do you think? So we're comparing Splenda, sweet and low, and equal. Whatever the highest number would be the least amount of artificial sweetener and the most amount of filling. So this would be, right, if you're going to equal 2 tablespoons, TBSP, tablespoons, right? So here I might only have, I don't know, a tiny amount of sucralose plus lots of filler, right? But what if I had something that was only 200 times more, right? Now I would have like 1/4 teaspoon plus some filler, right? So the more sweet it is, the less of the actual artificial sweetener you need. Make sense? To taste as sweet as 2 tablespoons. So this one should be Splenda. So if we did them in order, it would be Splenda, then what? Then sweet and low, then stevia, if you wanted to include stevia, and then equal. So it would go down this way, except for this one, which would be like here. Does that make sense? So this one, I don't know why they did that. They could have just done it one differently and it would have been fine, but okay, right? All right, kind of interesting, right? All right, polysaccharides. Polysaccharides have 2 really big functions generally, and that is storage. Like, you know, when you go to sleep at night, are you eating? Nope. So what's your body going to do? During the day, you've stored some sugars and you're going to break off one at a time and put it into the bloodstream so that your brain has a snack while you sleep, right? It's true. You're feeding your brain while you sleep. So that's one of the ways that we can use polysaccharides. Another major way that we use polysaccharides is in giving things structure, or giving things the ability to recognize a particular cell because it has this sugar on the outside of it, right? So let's talk about some of the big ones. Amylose and amylopectin. What do those 2 together make up? Starch. What is starch? It's a storage polysaccharide in plants. Okay. So describe the similarities and differences. So they're both a part of starch. They're both involved in storage. Storage of what? What do you say? Sugar, glucose. So they are both polysaccharides of glucose. Anybody have an idea of what kind of glucose? Does it have to be? D glucose. Okay, how are they different? Amylose, amylopectin. AMY. Come on. And you should have watched this video, so if you didn't watch the video, you're probably lost. So amylose. If they're both starch, they're both storage. You have another both? They both have alpha 1,4 glycosidic linkages. Lots of things that they're similar with, right? Okay, so, well, you're talking about the difference between the glycogen and the amylopectin, but you're on the right track because amylose itself is only a straight chain. So it's a straight chain, CHAIN. So basically what you have are these, my very, very simplistic drawing here, right? Over and over and over again, this is the same thing, right? This is a straight chain. Now, it's weird in that it's a straight chain that coils like a telephone cord. You know those old-timey telephone cords, like the one I got in my office? And it's got that little spiral in it. It's really a straight cord. They've just heated it and coiled it, right? So eventually, if you get it long enough, you'll get a coil going, but it's really a straight chain. Okay, so it's really an alpha 1,4 linkage. So that's amylose. Amylopectin is the same sort of deal where we have these alpha 1,4s, but what's different? It also branches off. So here, you're going to come up and you're going to make a glycosidic bond with another sugar molecule. And now look what I can do. I can keep going. I can add more glucose units. So why is this really important for us in general, for animals, plants, for living organisms? Why is it important to have a branch? So this has branches. So this is straight and branched. And the branched are they alpha 1,4s anymore? No, they are alpha 1,6s. So it's just attaching to a different carbon. So why do we like amylopectin? There's not just one branch, right? There are tons of branches. So if you only have a straight chain, I have to take individual glucose off one at a time, right? But if I have this big old ball, and you ever seen a Kush ball? Y'all, I'm marking my age. Y'all know what a Kush ball is? I'm so old. All right, a Kush ball, it's got like a core, and then it looks kind of like a porcupine, and then it's got all these little things pointing out, but it's soft, and you throw it at each other. I mean, you know, so you're not throwing a porcupine at each other. But so basically, that's what amylopectin is, in that it has all these branch points. So now you have all these different little fingers that are coming off of it that I can pull off one glucose molecule from every single end, and all of a sudden get a massive rush of sugar into the bloodstream, right? So it's very, very efficient, right? But the branches only occur about every 30 sugar units, glucose units. Glucose units. So you branch about every 30 glucose units. Okay. Oh my goodness, I got to fit this whole thing in here for amylopectin and glycogen. All right, so let's do some both things. Amylopectin and glycogen are very, very similar. They do the straight alpha 1,4 linkages. They do the branched alpha 1,6 linkages. They're both storage forms, right? Forms of polysaccharides of D-glucose. What's different? One's in animals. So amylopectin and glycogen. Glycogen is in animals. Amylopectin is in plants. It's not the only way that they're different. We're cooler. We're cooler. You want to know why we're cooler? We have more branch points. So we branch every about 12 glucose units. How many did we say plants do? Every about 30. So who has more branches, us or them? So we can move sugar into and out of storage more quickly than plants can. But they don't have to go anywhere, right? We kind of have to be able to get around. Okay. All right, describe the similarities and differences. Amylose and cellulose. What did we say amylose was in? Plants. How about cellulose? Both. Okay, so they're both in plants. Okay, what else could we say? They have no branching. Remember the amylose was the one that didn't branch? Well, cellulose doesn't branch either. So they're both straight chains. How about cellulose? This is one we haven't talked about yet. Where do we find cellulose? It's plants, but it's not there for storage. It's there for structure. So this is a structural, S-T-R-U-C-T, structural polysaccharide, right? It makes up their cell walls and things like that to give them that plant shape, right? They don't have bones, so what makes a leaf look like a leaf? Cellulose, okay? All right, what else? If it has a different function, can you bet it's structured a little bit differently? So it's also an alpha linkage, but anybody know what the linkage is? Oh, it's not an alpha. It's a beta. The ones that are strong tend to have beta linkages, and the ones that are for storage tend to have alpha linkages. So it's a beta 1,4. Whereas amylose, we already said above, was alpha 1,4, right? And we said amylose was there for storage. Okay, so now we've kind of talked about cellulose. What's the deal with chitin? Chitin is also structural, but it's not structural in plants. Anybody know what organism we could find it in? Bugs, crustaceans, right? They're there hard, makes up the exoskeleton. Makes up exoskeleton. And it actually has a unique sugar moiety. It has an N-acetyl glucosamine. You never have to spell that instead of a glucose. You don't have to spell it. Don't have to spell it. All right. I think that's it. And then we're only missing, oh, just a little bit. And then we have our blood types. So we'll be able to catch that up, no problem. All right, y'all have a good rest of your Wednesday. Yeah, I can't do it. Like, I would really love to stay in class right now and do it, but another professor comes in right after me. So I'm gonna uninstall it, reinstall it, beg someone off the street to like trial run it for me.
Don't you have a tech department here?
Do we?
No?
We do. All right, I'm gonna end the chat.